Hey, good morning. Good morning, California. Good morning, US, and good afternoon, Europe, and good evening, China, and the whole Asian countries. I'm excited to announce the inaugural event of Nanoscience Global Lecture. We know this is a special time for us to start this lecture series of Nanoscience Global Lectures. We indeed plan to hold this lecture series monthly. So each event will consist of 2 well-established speakers and 1 early career speaker. So the reason to start this lecture series, this is COVID-19. We believe it's very timely to initiate such a lecture series to bring the global nanoscience community together, to highlight the exciting nanoscience progress, and to stimulate the creative thinking to address the global challenges. The second is, this year is special for Nano Letters. We are now into the 20th years of anniversary. So in the past 20 years, Nano Letters has been serving the global science communities very well. We want to also highlight and also plan for the future. I'm particularly honored to announce the starting of this event. My name is Yi Cui. I'm a professor of material science engineering at Stanford University. I also serve as the executive editor of Nano Letters. So with these introductions, let me also introduce our chief editor of Nano Letters, Professor Terry Oldham of Northwestern University. She's a professor of chemistry there. Now let me hand it over to Terry. She will take over the introduction and moderate the lecture of our 3 speakers. Terry.
Great, thanks, E. And again, welcome everyone to our inaugural Nanoscience Global Lecture. As E mentioned, this is another start to celebrate the 20th anniversary of Nano Letters, which was founded by our first speaker, Paul Bostados. And there are also some other new things that I think are very special about the lecture. It's a direct collaborative effort between E and our early career board. They have been very interested in not only just promoting the journal, but providing an outlet to share the best nanoscience that is happening now. And I'm so glad that they initiated this and worked with E and partnered with this to make this event happen. It's really quite special. And I think what you'll hear today from the lecturers is some perspectives of what has happened over the past 20 years, what they're currently working on now, and the projection for the next 20 years. These are pioneers in the field. They have well-established track records of innovation and creativity. And we've also tried to balance some of the big ideas with emerging investigators. The emerging investigator in this series, first one is Laura Liu. She's already established in her own right, but she's just a little bit younger than these other 2 luminaries. And so we're excited to hear from her as well. So just a reminder of the schedule today. The first speaker will be Professor Paul Bostados. The second speaker will be Professor Laura Liu, and the final speaker of the session will be Professor Steve Chu. So you can type your questions in the Q&A, and then we will get to them after every speaker. And then finally, we'll wrap up with E moderating a panel discussion. Okay, so our first speaker is Professor Paul Bostados. He is the founding editor of Nano Letters. Thank you, Paul, for taking time out of your leadership leading during a pandemic and kicking off this amazing event. He is the University of California, Berkeley's Executive Vice Chancellor and Provost and Samsung Distinguished Professor of Nanoscience and Nanotechnology. His research accomplishments include studies of the scaling laws governing the optical, electrical, structural, and thermodynamic properties of nanocrystals. He has developed methods to synthesize size and shape controlled nanocrystals and developed methods for preparing branched, hollow, nested, and segmented nanocrystals. So Paul, I won't go through your exhaustive list of recognition for your science, and I'll just let you get to it.
Thank you so very much. Thank you, all of you. It's so wonderful to be with you this morning. I'm going to just start sharing my screen here. Well, I guess it's morning for me and different times of the day around the world for you. And I think we're in the midst of a global pandemic. We've been pulled apart. We're all remote. We can't meet together. And for so many of us, there are many other things that are such crises at the moment. We have a vast economic crisis, amplification of inequality that is also in many places creating crises of social justice. And here in California, at least, we've been living through the effects, the real effects of climate change with a series of fires that are just cataclysmic for our state. And so in the midst of all of that, as I thought about this presentation, rather than wanting to go through historical things, I decided what I would like to do with you is share what's happened inside my research group recently during a period when we were largely obliged not to be inside our laboratory in the normal way. We were under restriction in a shelter in place, so we could not be inside the lab. And we spent together, my coworkers and I, a time where we thought hard about how we could take advantage of some of the new tools that are emerging in data science and machine learning and artificial intelligence, and apply those to some of the problems that we have been studying for a long time inside our group. So I will try to take you through what has been something I would say of a joy for us during this period of enormous upheaval and difficulty. I hope from this presentation that you'll take away three things. I'm going to talk about how we know, all of us, that one of the foundations of nanoscience is to develop new kinds of experimental tools for visualization at the nanoscale, being able to see things. And for us in my research community, we've been working very hard for quite some time on the liquid cell transmission electron microscope, so we could visualize phenomena that take place in a fluid on the nanoscale with the resolution of electron microscopy. And there are many questions about that technique. It's still emerging. So what we'll see here is the use of emerging tools of data science, artificial intelligence, machine learning, in order to help us leverage this new tool and to help us make discoveries. And perhaps another theme I'd like to share with you is that although there are these many new tools that come because now we have new detectors, we can collect much more data, there are better kinds of sensors, we can store vast amounts of information, and there are also new methodological approaches to analyzing those. All of those are true. And yet still, for example, for the problems that we'll be talking about, much more classical approaches to science have to be integrated with those new tools in order for them to actually make sense and to lead to something that we really think is substantive and real. So I'll be showing three experiments today, but first I just want to remind that the liquid cell is a cell in which we trap a small layer of fluid, maybe 50, 100 nanometers thick, between two membranes that are electron transparent. They could be silicon nitride, or as often the case in our research, graphene layers of different thicknesses. And inside there, during a period of the last, let's say, 15 years that this technique has really been developing, since early work of Francis Ross. We started in my research group in 2008 in this area. So it's been a number of years now. We've been able over the years to look at things like the growth of nanoparticles, interaction and forces between them. Very often these particles are located close to a substrate because those are the ones that are immobile and can be seen quite readily, although there may be other particles flying around up above that that are not easily visualized. And so we've been able to observe many, many phenomena. But the question is, how does this become a more quantitative tool for nanoscience? And that's where some of these data science tools, I believe, will play a very big role. So first, let me just describe some studies of the motion of nanoparticles. Here you can see some trajectories of the motion. It's the simplest experiment possible. These are gold nanocrystals located near a silicon nitride substrate, and they're under illumination by the electron beam at different fluxes. Let me just put on my laser pointer here. You can see here, as we increase the flux of the electron beam from 2 electrons per angstrom squared per second up to 50, why then we see that if we always look for exactly the same time interval, we can see that initially the particles move hardly at all, although they do move, but not nearly as much. And then at a later time, their motion is much greater. And similarly, if we expand them all to fit, to take each one of these trajectories and make it kind of fill a similar area here, which is now very, very different time scales of observation and different, then what you'll see is that the actual pattern of the motion completely changes. They don't have the same character. And that's been known for a while, but it's actually very, very difficult. You can collect thousands of these trajectories for many, many, many particles. But actually taking each one of them and understanding them is quite a difficult project for human beings to do. Here are some basic models that could govern such phenomena. It could be, for example, that they simply display Brownian motion, although that doesn't seem like it could be true for all of the power fluxes that I showed you, simply because the whole shape of the thing seemed to change. But remember, in that case, if it's Gaussian, then we would have non-correlated steps. It's essentially random. If a particle goes in one direction, the next time it will go in another, and those steps will have certain sizes that are dictated by the thermal energy. We could have a situation where it's so-called fractional Brownian motion. There's something constraining the motion of the particle. There's something around it that won't give. So it can bounce around within a certain zone, but when it hits a boundary, it has to reverse, in which case, as you can imagine, when it reverses like that, many of the motions will be in the opposite direction from the previous one, and therefore it will have an anti-correlated motion. Or there could be a third version, in which we have somehow some very deep traps, and then other locations on the surface which are completely ones where the particle can move around much more freely. In which case we might have a situation where we would have a very heavy-tailed displacement in time and a heavy-tailed distribution of displacements. In other words, sometimes the particle would move very large distances. Sometimes it would stay in one spot for a very long time. So the motions will be non-correlated, but they will just have very odd, non-Gaussian-like characters to them. So those are three models that people have wondered about for many years. Do they apply? Do they not? We have a whole pile of these trajectories, which we've collected over many years, so we could go in and look at them. And if we do that, we can then create a type of neural network, a deep learning kind of neural network system, which will take vast numbers of these trajectories and look at how the motions correlate with each other at different time intervals after a particular observation. We can just see how do they correlate at later and later times with each other in a kind of very systematic way, and then classify the probability that every trajectory is either fractional Brownian, or continuous time random walk. And then we could do that as a function of the power of the beam or the electron beam flux. And so here you see an example of that, where we go from a very small flux to a very large flux. And what you see is that very clear data, I think, that it clearly changes from a case where we have fractional Brownian motion at the low fluxes, and we have a continuous time random walk at the higher doses, and that's an evolution that the system undergoes. This is we're still doing more of these as time goes on, but so far this is many hundreds of trajectories that are being analyzed in this way, and I think we'll be able to readily do many thousands. If we actually go in and look at the motions, we can, of course, see from particular ones the kinds of phenomena that I'm describing. In other words, what we can see, for example, is if we look at just the displacements as a function of the time on a log-log plot, this green curve is a Gaussian. I think you can see that it's really not quite Gaussian at the longer times, which is consistent with the fractional Brownian motion. It becomes a kind of power law at the higher fluxes, which is consistent with the heavy-tailed distributions. But the more important thing perhaps to look at is how the correlations evolve as we look to longer and longer time intervals. And what you can see is that they're clearly anti-correlated at the low fluxes, but they're non-correlated at the large fluxes. So if we put all of those things together, they're very consistent with this evolution of the mechanism. And if I now go in to try to speculate, now this is more speculative, and it takes me to our next topic, which is, so okay, we can learn this about it, but ultimately we have to understand the chemistry that's going on inside there in order to be able to have a microscopic model of this phenomenon. We hypothesize at this moment that there's a very dense network of hydrogen bonding near the surface at the low fluxes, which is creating a kind of gel-like network that produces the fractional Brownian motion at the low fluxes. And at higher fluxes, the actual creation of ionization phenomena and other things leads to a situation where there may be some local charged spots that are heavily charged and that lead to these kind of trap-like state. And there's a lot of reasons to think that could be true, but that's something that will now have to be demonstrated in future work. So the data science tool takes us a certain distance. It helps us pose questions and answer, get a sense of what the landscape looks like. But there's still much more to be done in order to understand the nature of what happens inside a liquid cell. Along those lines, the question always arises, what's the chemical environment inside a liquid cell? Because the electron beam is passing through, it can ionize things, it can create free radicals, it could do different things. And so this has been studied for many years, and Frances Ross has been involved in that for a long time at Nanotechnology. Beautiful work from her and from many others who have made numerical models of what the plain water exposure to an electron beam might look like. This would be the same with almost any kind of highly ionizing radiation passing through a very thin liquid cell. So we can draw upon the vast literature that exists out there about what happens when you expose water to different kinds of ionizing radiation. And in our case, we have developed over a period of about a decade, a kind of model system for studying this, which is to simply take gold nanoparticles of different shapes, different therefore energies of the facets, and place those inside an environment where they can be oxidized. And so it's always critical in these experiments to have present a species. Often we use iron halides like iron chloride, which actually chemically perform the oxidative etching. Now that then becomes a marker of the chemical potential. Is there enough oxidizing potential to oxidize the gold or some other species? And what I will show you today is that we have been able, Ivan Moreno-Hernandez has shown that we can now take a series of different redox couples, in other words, chemical species. We can place them inside the liquid cell, and won't be able to go through all of the experiments that show this, but effectively what happens is, depending on which chemical pair of redox potential pair we place inside the solution, it's the equivalent to setting the potentiostat on an electrode. And we can turn that up and down, allowing us to control the chemical phenomena that take place inside the liquid cell. Here you see a very simple example of that, where we have three redox pairs, VO2+, vanadium dioxide cation, chromium 3+, and cerium 3+. Each one of those can act as a pair inside a redox couple. And if we have gold particles present, what we can see is that we can vastly, vastly tune the rate at which the gold particle is oxidized. And so we can try now to rationalize that based upon some understanding of what those redox potentials could set the potential to be inside the liquid cell. And indeed, when we do that, we can find that, for example, we could have in this experiment here, two kinds of nanoparticle present at the same time, gold and platinum, and we can arrange the conditions such that, for example, the gold is oxidized very rapidly, but the platinum is completely left intact. Or we could change the conditions such that we are able to still etch the gold at the very highest potential that we can apply, which is with the cerium. Indeed, it becomes consistent that we set the oxidative potential higher and higher as we go from the vanadium oxide to the chromium 3+ to the cerium 3+, kind of like I showed you in the previous graph just with the gold. But we can put in many pairs of metals. I'm not going to show all of that today, many pairs of metals, and show which one can we etch, which one can we not, with different redox potentials. And if we go through all of those, I think in the end we can create a kind of self-consistent picture of how we can now set the chemical potential inside the liquid cell using chemical additives. This is incredibly important if we want to eventually be able to study much more delicate species inside the liquid cell. For example, biological matter. We want to be able to set the potential low in order to be able to observe that. Or we may have a case where we really wish to set it higher in order to achieve a particular goal that we may have. So for us, this has been a kind of milestone event in some sense of being able suddenly to have the ability to set the potential inside the cell. We can demonstrate this, for example, sorry about the graphics not quite working here right, but we can set that, for example, by having a binary particle that has two different components, and depending on which redox couple we place inside, we can etch one and not the other. So that's an important development for us. And I think you can see that very clearly in the evolution of the shape here of this binary nanoparticle. Now, remaining question then is what happens when we have just plain iron there, which we've studied for many, many years. And with iron present, we can, of course, etch the nanoparticle. Does that fit in with this model that I've just shown you, where we set many different redox potentials depending on which redox species are present? Does the iron fit that story or not? And honestly, at first, the story is it appears not to, because the iron 3+ itself that we put in when we put in iron chloride doesn't have enough potential by itself. If you just look up in an electrochemical table, it doesn't have enough potential to actually perform the oxidation of gold. So what gives? How is this actually working? So let me just show you some basic phenomenology first. In the case of, if we have no iron present, there's absolutely no etching. Okay, that's good, because we think we do need some kind of oxidative species to be generated inside the liquid cell. And we can observe phenomenologically that if we put in different halides, chloride, bromide, iodide, the etching rate changes enormously. It goes from a very modest etch with the chloride to a very fast etch with the iodide. You can see how fast it is because it completely starts tearing layers off the particle very, very quickly and making these shapes that are extremely rough when we have too fast of an etch rate with the iron. So we know that iron cations are required and that the halide ions play a role, but is it the halide network of radical species, because halides could generate radical species, or is it some other mechanism? And it turns out it is some other mechanism for the halides, and that the iron cations themselves are necessary components. We can see that here in some studies where we perform collaborative work with Alex Kant and Wolfgang Nolle at the Leibniz Institute of Surface Engineering. Kristen Alby, a postdoc in my group, has helped us forge this collaboration. And in this situation, we can see that there's, if we do a pulse radiolysis study of a bulk volume of liquid that has iron chloride inside of it, we can see a very clear signature of the presence of iron 3+ decaying very, very quickly, and actually creating present some other long-lived species that absorbs light at different energies at different timescales, all of which is very consistent with the notion that the iron 3+ is oxidized to a higher oxidation state, and that's a long-lived species. And that species is the one which actually tears the gold apart and sets the potential for the etching when iron chloride is present. The exact identification of that species, well, okay, we could debate it a little bit. I'm not going to say it today, but we're working on trying to home in on it and get to know it. One thing I can say is that we can prove that a very long-lived species is responsible, because what we can do is start the etch of an iron particle in the presence of a gold particle in the presence of iron chloride, and just turn the beam off, or turn the beam down by a massive amount, and then observe that the etching continues. That would never happen at such a low dose rate if there were not a long-lived species present. So that kind of is something that we can demonstrate pretty clearly. And so this is our current model of what goes on when we just have iron present. That is, that the iron is created into a much more highly oxidized species, which is responsible for atom by atom removing golds. but that the reason that the halides are so much more active when you go from chloride to iodide is actually because of the equilibrium constant of the binding of the halides to the gold. They're essentially pulling those gold atoms out, and that's how that works. So that's kind of our picture right now. And I guess, you know, what I would say is this is all kind of standard chemistry stuff, even in this era of data science, but it's helping us very slowly to pull through a picture of what's the chemistry like inside a liquid cell? Can we control it in such a way that in the end, whichever sample we wish to study, we would know the chemical environment that we're investigating. And I would say compared to even just 3-4 years ago, the progress in this area is enormous. And I'm feeling quite good that within a few years' time, the liquid cell can be used with much more confidence about what the chemical conditions are inside the cell. So we've also been able to do this recently with some very, very sensitive components like lead chalcogenides, which are easy to see in the electron microscope. I'm not going to go into the details of that, but I will say that we can set the potential low enough that what previously, if we had tried to observe these, they just disappeared so fast you couldn't make it work. But now we can set the potential low. It's kind of a marker that we can really set potentials low inside these systems. Okay, I'm going to share one more last piece from our spring and summer of data science playing around. And this is a case where we've just done something that I think is a little bit needed inside the nanoparticle business. We, of course, always are collecting electron micrographs of samples, and we try to make them one shape. But the question is, are they really all one shape? And how do you actually know? How could you actually be as statistically precise about whether all your particles are the same or different from each other? How could we classify them without presupposing what the answer is based on a first impression that we may get by looking at a selection of the sample? So in this case, Ringjie Wang has taken a method of, he's developed a method of unsupervised. In other words, we do not tell the algorithm anything. It's a machine learning algorithm that's unsupervised, that spontaneously locates and classifies particles. It looks at every particle that has a density change in it, as you can imagine, and it finds the boundaries of those. In this case, we're only looking at convex particles, although perhaps in future we could do concave ones. And we just classify every object that's detected that way according to its area, its eccentricity, its aspect ratio, its circularity, its solidity, and its convexity. So that's a whole bunch of parameters there, actually 6, and that's collected for every single particle. And then all of those are looked at to find how many independent classes of particles are there statistically present inside the system. So for example, here's an application of this in which you can see that we took 2 kinds of gold nanorods. Both of these preparations in the literature are thought to be ones that create rather pure preparations of nanorods. And of course, if you go and look at some parts of the micrograph, you will find ones where it looks like it's 2 kinds of, and maybe, okay, there's one out there that's an outlier. But if you go to other regions and you look around hard enough, you'll find more that don't quite look like that. And in fact, if you look at this classifier method, you'll see that it really does identify 3 classes of particles, not 2. You can see them all kind of stacked up here, and you can see that there are these 2 that are the vast majority, but there is this smaller amount here of this other shape, and that's present inside these syntheses. Indeed, if you go through and look at the different classifiers, you can see that if you only looked at one of these parameters, you wouldn't really be able very easily to create a good classification. It's by looking at all of them and making sure that they are self-consistent with each other that you can actually find out what's present. And it, you know, this does quite well if we actually ask people who have already now know the fact that there are 3 kinds to go through and find them. They do. I'm not sure they would have done this spontaneously because this one's present, and sometimes you don't see it because it's kind of in a different part of the sample zone. So, you know, it's a tricky problem in analysis. What we're saying is that you could go through now and just collect vast amounts of area and then go and classify it somewhat objectively. Here's a second example where we've taken a synthesis that's very well known to produce beautiful triangular prisms, and we've applied the same thing. And at first it kind of localizes and says, well, there's 2 kinds of particle. But now, if you actually go in and look at these 2 kinds of particle, what you can see is that there are ones that have almost very sharp corners like this one here. And then there are ones that seem to have far more rounded ones. And if you keep going on that, they seem to get more and more rounded. So we take this classification into two kinds of particle, ones that are something happened unusual in the synthesis. It's definitely a minority, but it's present. And then we take this and apply our algorithm a second time to it. Why then it does again classify it into different groups. And in fact, it's interesting because this time it actually finds that there is some distribution here. There is really a much more triangular component, but there's another component that has a kind of a greater degree of almost hexagonality to it, if you like. And if we kind of go through and look at that, what you can see is that there are particles that are indeed extremely triangular, and then there are particles that really, if you look here and you hunt a little bit, you'll see ones that look quite hexagonal. And there's kind of statistical distribution of those all across that classification range. And I think what you can see here, what this algorithm will allow us ultimately to do, is to try to understand why this happens inside this sample. I think what's happening is that the energy penalty for, there is an energy difference between these. If you imagine this one as being, well, let's say in this case, if it were a perfect hexagon, every side as you go around the 6 sides would be equal in length. And imagine now changing that so that they alternate and every other side is shorter and than the previous. Then the energy, of course, will change. You're creating tips. You're going from flatter surfaces to more tipped ones. And the energy will change a little bit as you do that, but the synthetic conditions may favor one facet over another, or may favor the formation of those tips, or that could be a kinetic factor. You can debate it different ways, but what you can see here, this is actually a distribution of how the shape changes across the sample when you collect large numbers of these things. And we could have done this previously, perhaps, but this is something that allows, again, to happen at vast scales. You could do this with huge numbers of samples and really get data that I think is, good to be able to access in this way. And this will allow us to look at this transition zone and perhaps to come to understand what are the kinetic and thermodynamic factors that lead to the width of this zone. Can we push it around a little bit? Can we manipulate it? And so on. So I'm very pleased that we have this method now. I think we're going to use it all throughout our research group, and we're deep into creating versions of it now which correlate with other properties that can be measured on the same samples in the same way, optical properties and others that would be sort of correlated between these two. I'm going to just take a moment now and wrap up. I think the theme of what I've tried to tell you is that there's this the advent of new detectors, new visualization techniques, in our case, the liquid cell electron microscope, is that it is a very powerful tool. And when all of these new tools of visualization will be enhanced massively when we learn more in our community, the nanoscience community, how to take care and use the data science tools, also robotics, many other things like that. But as we are able now suddenly to collect vast amounts of data and also to perform these new computational tools, the marriage of those two creates an environment where we can interrogate our samples in different ways, and where the tools of deep learning and machine learning and others of that kind will allow us to ask and answer questions which previously we could get at, but just in bits and pieces, because human beings trying to do them in small numbers just couldn't always do enough of them. At the same time, I want to balance that out by saying that that kind of work will work best when it's integrated deeply with a kind of trying to understand the fundamental physics or chemistry that kind of govern the system that you're looking at. In our case, here's in the middle here with Ivan, a very simple concept of redox potential being used to control the chemical environment inside the liquid cell. And so after a journey of many people, I think we're getting to the point where the liquid cell itself, not only will we be able to understand the mechanisms that are happening inside there, but we'll also be able to control the environment well enough that we could have some confidence in being able to say, well, when we observe this phenomenon in the liquid cell, this is the underlying conditions under which it's occurring. It's not a black box anymore. That's emerging, I think, in a very clear way. I'd like to thank these people. Vida Jamali did the work on the trajectories, the fractional Brownian motion and continuous time random walk. Michelle Cook has been doing the work on the unraveling the mechanism of the iron chloride, and Ivan, I mentioned already, the redox potentials, and Xingji has been doing the work on the machine learning for analyzing nanoparticles, and all of these folks have contributed in different ways to some of those projects. And I'm going to wrap up there and say thank you.
Great, thanks, Professor Bostados. We have many questions coming in. As a reminder, please put them in the Q&A and not the chat. It's easier for us to organize them in that way. So the first question is related to the beginning of your talk. And the question is not gold or platinum, these are precious metals, is this selective etching difficult for commercial research? Why do you not focus on low cost metals for commercial applications?
Well, in this case, we're using those metals in order to provide an understanding. In those experiments, the etching is of the gold or of the platinum or palladium and so on. The role that those metals are playing for us is as entities that we can describe very easily in terms of their chemistry. I didn't really go into this because it's prior published work, but we have shown in previous work with the iron chloride that we can in various ways set the rate of etching, not the chemical potential, but the rate. We can set the rate at which etching occurs. And when we do that, we can compare it to a model. The model is one where we have the ability to know what energy is required to pull a gold atom off from a theoretical model. And depending on whether it's coordinated to 6 or 5 or 4 atoms nearby, depending on what surface it's on and so on, the rate will vary enormously. So in previously published work, I didn't reference today too much, we've shown that we can really use very, very simple models to explain the rates at which we etch these materials. And so we're using those noble metals because they can be explained, the phenomena can be explained with a pretty straightforward model that gives us confidence that then we're using the metals to probe the liquid environment. Because then once we really know the liquid environment, what its chemical potential is, then we can use it to study anything. If you want to study corrosion, or you want to study the evolution of a battery component, or any other practical thing, you need to have that knowledge first. Well, you don't need to, but it sure makes it easier. So that's the reason.
Great, thank you. Just a short follow-up question on this. What is the role of the electron beam in these redox reactions? How does it affect the overall system?
So the main way in which the electron beam is affecting things is by generating a set of radicals. Those are the radicals that have been well studied in many radiolysis experiments over the years, and which are present, for example, in the Schneider and Ross models that I showed. So there's a kind of radical soup that's inside there. And what I'm explaining today is that radical soup, its actual chemical potential can be manipulated by chemical additives. When you may add something to it, you actually can adjust its chemical potential, its effective oxidizing potential up and down. So the electron beam does have that effect. It does not heat things very much at all. The thermal conductivity of the membranes is quite high. So things are not actually getting hot inside there, but they are, of course, having a situation where there's a large number of radical species present. And I didn't go into it today, but for each one of these systems, we have a model with hundreds of chemical reactions going on inside it that's based on what has been learned by individual chemical reactions in post-radiolysis studies over many decades. So there's kind of pretty good model of what the chemical environment looks like when you don't add redox species. What we're trying to do is add on top of that what happens when you add a redox species.
Great, thank you. This concerns the later part of your talk. Have you observed in the nanoparticles unusual phase structures that have not been observed in the bulk?
We've been looking at pretty simple systems so far. The answer to that's no. I mean, throughout the study of nanocrystals, there are a number of examples where people see, rather different structures in small sizes. But in this case, we're not actually looking at systems like that. We're just looking at some pretty simple metals. Just for calibration of these methods, but it's a good question. I mean, there are absolutely examples where phases are stable in finite size that would not be either because of the surface energies or because of other electronic phenomena that occur in small size that can stabilize one over another, but we've not been doing that recently anyway. Yeah.
In the machine learning part of your talk, can triangular antiprisms be differentiated from triangular prisms, or these different symmetries of three-dimensional structures?
Yeah, I'm sure we could. I mean, the one thing we can't do so far, I think I mentioned briefly, which is to look at concave particles. It just turns out that the algorithm that we've been using so far doesn't handle that very well. And as you may know, there's lots of funny shapes that are concave. For example, I mean, we've studied over many years tetrapods, or many other shapes that are, that have, when you project them down onto a two-dimensional plane from the image, they have pieces that curve the other way. And our algorithm at present doesn't really work for that, but I think we'll probably get to one. And that would allow us to look at more different kinds of shapes. But yeah, I think we could look at almost any kind of concave shape. This thing is going to pull it out pretty well.
And then the final question, where do you see opportunities for predictive modeling describing nanoparticle interactions and functions? How do you see the integration of mechanistic and data-driven models in this?
Oh my gosh, I think that's a very rich area. I briefly flashed on the screen some earlier work where we have studied the interaction potentials between nanoparticles in the liquid cell. We can observe how the trajectory of one particle influences another. And those are, of course, the forces which are at work during self-assembly in so many ways. So I think the liquid cell, it's possible to study drying inside the liquid cell and observe crystallization of nanoparticles in that way. It's a very, very rich area. Beautiful work I saw this morning in Nature magazine of this week from David Pine, if you haven't seen it yet, of making a tetrahedrally bonded, diamond-like structure with some colloidal particles. I think the area of self-assembly, the forces between particles can readily be studied with the liquid cell methods. And I think that's a very rich area for working between theory and experiment, because all of the models of self-assembly, of course, rely upon an understanding of the forces. So being able to try to visualize those is a big deal. And I think that's going to get better and better as time goes on. And I do think the assembly of nanoparticles into arrays of artificial atoms, into artificial solids, is a wonderful, beautiful area. I didn't get a chance to talk about it today, but we've been doing lots of work where we try to make oriented attachment in periodic arrays of nanoparticles in the last few years, and that's going really well. That's a rich area, and it's really worth studying.
Great, thank you. Well, we have to stop here to move on to our next speaker, but thank you, Professor Bostados, for being our inaugural talk.
Thank you for allowing me to be with you today.
Okay, so our next speaker is Laura Nalu. She is the director of the Second Physics Institute at the University of Stuttgart, where she also received her PhD. She works at the interface between nanophotonics, biology, and chemistry. Her group focuses on developing sophisticated and smart optical nanosystems for answering structural biology questions, as well as catalytic chemistry questions in local environments. Professor Liu serves on the editorial advisory board of multiple journals, including Nano Letters. She was also the 2019 Nano Letters Young Investigator Lectureship Award winner, whose lectures had to be delayed multiple years, but now she is finally able to give just a small piece of it. So Laura.
Thank you very much, Terry. Good morning, everyone. First, I would like to thank also Professor Cui and also the Nano Letters team for inviting me to present our work. I'm going to talk about DNA origami nanotechnology for dynamic plasmonics. So the field of structured DNA nanotechnology could be traced back to the original idea of Professor Seaman at New York University in 1982. So he had wanted to organize proteins in 3D so that he could study these structures using X-ray crystallography. So nearly after 40 years of research effort, DNA has been widely used as building blocks for nanoconstruction. So then the breakthrough came with the concept called DNA origami, and that was demonstrated first by Paul Rosamond in 2006. The basic idea is to fold DNA into nanoscale shapes and patterns arbitrarily. But the formation of DNA origami is as follows. So it involves the folding of a long single-stranded DNA molecule like a loop called M13 by hundreds of these shorter DNA strands into 2D or 3D arbitrary shapes. So origami is a Japanese word meaning paper folding. Therefore, the folded structure is often called DNA origami. The key to functionality is these capture strands that we can extend from the origami template, because they can provide sequence-dependent surface tags for assembly of functionalized entities together, including metallic nanoparticles, quantum dots, dye molecules, among others. Importantly, they also provide addressable bending sites on the nanometer scale accuracy. So this is very important for nanoconstruction because they can provide feature sizes much smaller than those obtained from top-down nanotechniques. For instance, electron beam lithography, focused ion beam etching, etc. So then in this talk, I will be very pleased to show you a number of DNA-based dynamic plasmonic nanosystems with tailored optical functionalities. So the question is, how do we build DNA-based artificial nanosystems with tailored optical functionalities? So if we look into living cells, actually there are many molecular motors which can perform very complex mechanical tasks. For instance, you see on the left, this rotary motor ATP synthesis. It can perform the rotating motion, efficiently pumping protons against their electrochemical gradient. There are also linear motors, for instance, kinesin 1, which can walk stepwise along microtubules and deliver proteins as cargoes for intracellular transport. There are also sliding motors. Myosin and kinesin 5 can slide along actin filaments and microtubules, respectively. Then we were inspired by such natural wonders. The first system we have built was a dynamic system with rotation, and that was done by my previous postdoc, Anton Kuzik, who is a professor in Finland. So we deliberately chose to build a plasmonic Carroll system because we know that circular diacoism spectroscopy is very sensitive to three-dimensional conformation changes of Carroll molecules. So in this case, we have this cross-like origami template. It has two origami bundles connected through their hinge area. And then we assemble two gauge nanorods on two bundles respectively, and they are actually functionalized on the surfaces with DNA strands. And then we extend complementary DNA strands from the origami bundles. In the end, due to DNA hybridization, they can be assembled very accurately on the origami template. So we built both the left-handed version as well as the right-handed version. Due to the fact that while light interacts with such a plasmonic system, then plasmons can be excited in these two gold nanorods due to close proximity of the two gold nanorods, they do couple. So then if we change the angle between these two gold nanorods driven by the DNA-based molecular processes, then we can change the configuration of such a plasmonic system so that we can track the optical signal changes in real time. The molecular processes we have been using are called Toehold-mediated strand displacement reactions. So basically, if we have 3 DNA strands in 3 different colors, so originally this blue strand is partially hybridized with the green strand. Then if I add the red strand into the solution, it can gradually displace the blue strand and fully hybridized with the green strand. So this is the operation schematic. You don't see the gold nanorods. I just want to make the image look simpler. So originally, these 2 locks are open on each, so on both sides. And then for the first Toehold mediated strand displacement, this double-stranded DNA molecule on this arm can be unzipped. And now the 2 blue parts here can be locked through DNA hybridization. Then the system is locked to the left-handed state. If we add return strand through the second Toehold-mediated strand displacement, this lock can be opened. Then the system can go back to the relaxed state. Similarly, we can also run the system between the relaxed state and the right-handed state. Just give you some impression about the working principle. You see, we add the DNA driving strands into the solution, and then this DNA lock can be opened or closed, depending on what kind of sequence-specific DNA strands we add into the system. These are the TEM images. On the left, you see the origami frames. And then on the right, you see how gold nanorods are assembled on these origami frames. Then we did optical tracking. Maybe we take a first look at the right figure. So here, the CD spectra in different colors, they correspond to left-handed signal, relaxed state, and then right-handed state. Then we tracked the optical signal at fixed wavelengths around 750 nanometers. Then by adding alternatively the DNA driving strands into the solution, then we can see or identify very clearly 3 different states, namely left-handed state, relaxed state, and right-handed state. So we do see system degradation over time because we needed to always add DNA strands into the solution. Then we figured out to also build a light-driven plasmonic nanosystem because we know light is a very clean energy source. It doesn't produce any waste in the system. So we have this ozobenzene modified DNA lock in red, you see here. So basically, it can be opened or closed in response to UV and visible light alternatively. So in the sense that we can translate light-induced molecular motion of ozobenzene molecules into much larger optical signal changes in a very robust way. So over the last several years, we have tried really different dynamic controls to really operate our plasmonic systems. I have briefly shown you the DNA control, light control. We actually also have implemented pH control, temperature control, aptamer control, among others. So I'm not going into the details of different controls because maybe you have realized until now, I have only demonstrated to you the switching among several angles. You might wonder, so where is the full rotation, like the case of this ATP synthesis machine, which can do the full rotation. So indeed, very recently, my postdoc, Dr. Ling Xin, has realized a plasmonic nanoclock. So in this case, this rotor gold nanorod can rotate against the stator gold nanorod with full rotations, clockwise or counterclockwise. I briefly show you the design schematic. So in this plot, you see this ring track. So this ring track actually has 16 bending sites in 8 pairs. So basically this bundle, which is used to host this top gold nanorod, can be released from one pair of so to say the footholds 1 and 1 prime, and then it can be attached to the next available footholds pair. So namely 2 and 2 prime. So in this case, when we add the corresponding driving strands into the solution, then this central scaffold strand can always hold this top bundle when it does the release and attach processes. So every time it steps forward with a rotation angle pi over 8. This is very similar to the case, if you go to an airport, you see such air traffic tower, and this top one is this air traffic antenna. It rotates clockwise or counterclockwise against the tower base. It's very similar to our system. We have these 2 gold nanorods, and then one is rotating clockwise or counterclockwise. Then we did the experimental tracking. And we have 2 colors to identify the counterclockwise rotation and clockwise rotation. So one sees that at different angles, so these 2 sides of data points, they agree with each other very well. It means that the system is fully reversible. So we have also tried another exciting version. So that is autonomous rotation. So that means we don't need to input the DNA driving strands into the solution. So rather, we can let the system rotate autonomously. So in this case, we have the foot, which is a DNA enzyme. So basically along the track, we have RNA substrates. The DNA enzyme can catalyze the cleavage of the RNA substrates in a very controlled fashion. And then in the end, we could also use CD spectroscopy to track such autonomous rotations counterclockwise or clockwise. So I'm not going to elaborate this aspect. So rather, I save time to show you the second example. The second example is a dynamic system with walking. So I have briefly showed you this molecular walker, which can literally perform stepwise walking along microtubules. So it carries this gigantic cargo and delivers it to different target destinations in living cells. We have built a plasmonic version. We call it Plasmonic Walker. So this Walker nanorod can walk along this origami track stepwise. Every time it does a 7 nanometer step forward or backward. So it does the transduction of nanoscale motion into change of optical response in real time. And this work was done by my previous postdoc, Chao Zhou. So in this case, this Walker gold nanorod in yellow, it does the 7 nanometer step forward or backward. So one knows that 7 nanometers is well below the optical diffraction limit. So we were wondering whether we could really use standard optical spectroscopy to track these nano step changes in real time. This is the walking principle. So we add the DNA driving strands into the solution. The foot is released from the original position, and then the new foot is bound to the next station. Then it can perform a 7 nanometer step forward. This is the experimental tracking. The walker goes from the left configuration to a arrow, and then it arrives at the right-handed configuration. And just to give you the impression of the CD spectra for different stations, although the distance difference is only 7 nanometers between neighboring stations, one sees that here the CD spectra actually are very distinct from each other. Then we monitored the CD signal at around 680 nanometers, so we do observe the nanostep changes, and we can even identify the walking directions, forward or backward, optically. All right, this is the next example, and that is also the last example I would like to elaborate. That is a dynamic system with sliding. So here, this what I'm showing you is this mitosis process. So it is a part of the cell cycle. When replicated, chromosomes are separated into 2 nuclei. So kinesin 5 is such a plus-end directed motor protein, which slide 2 anti-parallel microtubules apart for segregation of chromosomes along opposite directions. So if one sees the details on the left, so this kinesin 5 protein crosslinks and slide doublet microtubules powered by ATP hydrolysis. Then in our case, we have built an artificial analog. We have 2 gold nanocrystals which crosslink and slide doublet origami filaments powered by DNA hybridization. This work was done by my previous PhD student, Max Ubang. In order to track the sliding dynamics, we have also positioned a Fred pair on one side of our slider system. So basically it has a donor and an acceptor. And when it does sliding, these 2 gold nanocrystals can work collectively together to slide the origami filaments back and forth along 2 opposite directions. I show you also the TEM images. On the left, you see the initial state. So here, this corner shows you the reconstructed image from many of those structures in the background. So it shows this the symmetric configuration before sliding. And then after 2 sliding steps, it means that it introduces 28 nanometers displacement. And one does see this asymmetric configuration that is formed. Then we did the optical monitoring of the sliding dynamics. In total, we have 5 sliding steps. This is the initial state. There is no displacement, and then it can slide to the left or to the right. We also did the fluorescence intensity tracking as well as the donor tracking. So basically, we can identify very well the sliding steps optically. So we also did the theoretical calculations. It is worth mentioning that, so here in the model, we included the Fred effects as well as the quenching effects. Because here, when we do the sliding, when the slider does the sliding, the distance between the 2 dimolecules is changing at certain states. For instance, here, then you see that the donor is also closer and closer to one of the gold nanocrystals, and it introduces the quenching effect. By including both Fred and quenching effects, we found that the agreement between the experiment and the calculation is pretty good. All right, I'm going to summarize. In this talk, I tried to convince you that by the combination of DNA nanotechnology and nano-optics, it is possible to translate nanoscale motion into tailored optical functionalities. So this is very exciting for nanophotonics, as it really opens a pathway to build functional nanophotonic devices with addressability and programmability on the subwavelength pixel level. All right, so in the end, of course, I would like to thank all my students for their hard work and the funding agencies for their financial support. And I would also like to thank you so much for your kind attention. Thank you.
Thank you for the terrific talk. We have many questions, and a great time for discussion. So the first question is, thanks for a great talk, Dr. Nalu. I was wondering if you have been working with smaller nanorods. And in that regard, are there limits to the size of nanorods that can couple to DNA origami? And then does the diameter require a minimum size to have enough DNA loading for it to be able to hybridize with the handles on the DNA?
So the thing is that in our work, what we have been doing is that we really want to correlate the nanoscale motion with optical signals. So in the sense that we need substantial size of the nanoparticles to see strong optical signals. So that means we actually prefer to have larger nanoparticles so that they can provide strong signals for us to track.
Okay, next question is very interesting work. I just was wondering how fast the clock can rotate or slide, and how much load or weight can it take for rotating or sliding? What parameter can be used to modulate the rotation speed and capacity to take the load?
So this is a very valid question. So for different systems we have tried out, they have different speeds or switching speeds. So just give you some examples for the DNA driven systems. So they are slow. So the switching speed is on the order of several seconds. And then we also tried, for example, light control. That was faster, and then pH control was even much faster on the order of several seconds. I would say the most promising, from my opinion, approach to control such DNA-based optical systems is the electrical control, because DNA origami is also charged, and then they can respond to electrical control very well. I would say then in the end, we are quite confident that we can achieve a switching rate on the order of several milliseconds. And that work actually has been pioneered by Frisch Simmel in Munich with the electrical control of origami structures.
The next question is, thank you for the very interesting system, which has technical significance. I am wondering what would be some of the potential practical applications of these systems?
So from my opinion, I think that is because we have been building different dynamic systems, and then we use optical signals to correlate the nanoscale motion. So from my opinion, I think the very rewarding and really exciting direction is really the robotic drug delivery, because efficient delivery and precise release of drug molecules are of really great importance for our lives and society. So in fact, the researchers in China, Professor Ding's group in Beijing, these researchers have pioneered the field of robotic drug deliveries, because DNA origami can work as carriers to encapsulate drug molecules, and also they can be functionalized with aptamer locks that can be opened when they recognize the proteins expressed on the surfaces of tumor cells. So in the sense that with such intelligent drug carriers, then we can really significantly enhance the efficiency and accuracy of drug molecules. Therefore, I think this is a very rewarding direction from the field of DNA origami technology, so which deserves more research efforts.
The next question is, thank you for the nice talk. I was wondering how much control you have over the motion and the effects of pH and the surrounding system-like temperature.
So in fact, for the system I have shown to you, we have optimized the system for a long time, and then they could work efficiently. So pH is a very important parameter that we have to really take care of for each experiment. So we run the system roughly at a pH value around 8, but for pH controlled systems, of course, we could also tune the pH range for a certain range for different responses. So in the sense that for each experiment, one needs to perform a series of parameter studies until the efficiency and productivity are satisfying, I would say.
And the final question, there's still some others, but based on time, is what is the control over the reproducibility of the clock motion at specific rotation settings?
Mm-hmm. So for that rotor system, it was really very challenging for us to implement because it is a very complex structure, first of all. And then we also had several versions before the final version. So then in the end, I think the yield was, the structure yield, I think it was on the order of more than 50%. So we did everything in bulk. And that was the challenging part. So what we have been doing with our collaborator in Munich is to really track the rotation on a single structure level. Then in the case that we can even understand the dynamics of the rotation much better because we don't have the ensemble effect.
Great, thank you very much. Let's thank
you.
Professor Nalu again for this terrific talk.
Thank you.
We will now transition to the final speaker of this first inaugural event, Professor Steve Chu. Steve Chu is the William R. Kennan Jr. Professor of Physics and Molecular and Cellular Physiology in the Medical School at Stanford University. He is the former Secretary of the U.S. Department of Energy. He is the co-recipient of the 1997 Nobel Prize in Physics for his contributions to laser cooling and atom trapping. And he has published over 280 papers in atomic and polymer physics, biophysics, biology, bioimaging, batteries, and other energy technologies. Welcome, Professor Chu.
I'm going to unmute myself and thank you very much. I'm allowed to share my screen and let me just see if this works. All right, can you see the talk? Yes, great. All right, I'm going to talk of a few selected topics out of many of how nanoscience can actually spur on innovations, and it ranges from climate change solutions to understanding molecular motors, not the kind you just heard about, the artificial motors, but the ones that nature has given us. So let me just set the stage by telling you one or two facts that I hope you all know. Currently, the amount of CO2 in the atmosphere is higher today than it's been in the last 23 million years. And if we look back 23 million years ago, the Earth was about 3 or 4 degrees warmer. And the other thing you should know is there's about 100-year time delay from what we've already done, the damage we've already done, to what we'll actually see. And to help you understand that, think of times, perhaps older people know this better than younger people, when they got a lot of sun, they used to sunbathe and get tans. And today they have lots of skin cancer. You don't get skin cancer the first time you get sunburned. It takes 10, 20, 30, 50 years to build up the mutations. Similarly, if you start smoking, we know that smoking in many people leads to heart disease, stroke, and lung cancer. Again, you don't get these diseases your first pack of cigarettes. It takes, again, decades to do this. And so we have a similar situation with what's happening with the climate. in order to keep the temperature below 2 degrees, the climate scientists have estimated that you have to stay below about 3 gigatons, 3 billion tons of carbon dioxide of cumulative emissions. Why cumulative? Because once you put the carbon dioxide up in the air, about half of it stays up there. It circulates between land and ocean and back to the atmosphere. About half of it stays there for 10,000 years. So it's not as though you stop acid rain in about 10 years, the lakes and streams clear up. This is a very, very long-term effect. And so this is the worldwide cumulative emissions. You see that most of the cumulative emissions occurred over the last 65 years. And the rate we're going will exceed this danger line in 14 years. The danger line of 14 years is about 450 parts per million. I unfortunately think we're going to be going over 550 parts per million. All right, so there's an expression that Yogi Berra might have said, but he didn't. If you don't change direction, you will end up where you're heading. And this is where we're heading. And the other thing, as a history lesson, not as a climate prediction, in a world just 2 degrees warmer, the sea level was 6 to 9 meters higher than today. And we didn't know this from the fossil record. Okay, so we've got to transition to cleaner solutions. And one of my favorite quotes was said by Sheikh Yamani, who's former Saudi oil minister. And he said, the Stone Age came to an end, not for lack of stones. And the oil age will end, but not for lack of oil. And what he meant by that is we transition to better solutions. You don't look at stones underground and said, you know, if it weren't for politicians and regulators, we wouldn't have these stranded assets. You don't look at stones on the ground as a stranded asset. And so the corollary to this is if you don't find a better solution, oil and gas will be used. It's going to come out of the ground because there's trillions of dollars of stuff under the ground. So you've got to replace it with a better solution. So let me tell you about very promising developments in electric vehicles. It's only a small sliver of a solution, but it's still important. Here's a prediction from Bloomberg New Energy Finance of how electric vehicles down here in the purple, BEVs, or plug-in hybrids in the blue, will gain in market share. And they predict by 2040, over half of all the light car sales will be electric vehicles. That's the good news. The bad news is about 45% of the new car sales will still be internal combustion engines. And those cars will last another 15 years. So actually replacing the automobile fleet in a half a century, it would be considered actually moving quite fast. So everything goes slowly. All right, now let me show you a few pictures. This is Easter morning, 1900, the Easter parade. This is Easter morning, 1913, New York City, Fifth Avenue. And what's the difference between the left-hand picture and the right-hand picture? Well, in the left-hand picture, there's one horseless carriage, an automobile. On the right-hand picture, there's one horse-drawn carriage. And so what we've seen is a transition that went much, much faster than electric vehicles. That transition required huge infrastructure. The transition to automobiles required gasoline oil supply chains, paved roads, repair shops, the whole works. But there was a serious environmental pollution hazard that actually hastened the transition. And that pollution was the fact that in 1880, New York City had 160,000 horses. Those horses were producing 3 to 4 million pounds of horse poop a day. And it was piling up everywhere. And you couldn't help but see it and smell it. And it was that pollution that actually did something, and that really moved the needle in cities. Here's another example of pollution. This is Beijing and a bad air day. And what we found in the last decades is that particulate matter, the very fine PM2.5, the same size as cigarette smoke, is particularly deadly. And large epidemiology studies suggest that 5 micrograms of PM2.5 are enough to increase your chance of getting lung cancer by 18%. And this was based on following over 300,000 people for nearly 13 years. The average air in Beijing in 2016 was 76 micrograms per cubic meter. In California during the forest fires, it was well over 200 micrograms per cubic meter. And the World Health Organization guideline is actually for clean air is actually 10 micrograms per cubic meter. And this is causing some consternation because if you consider clean air giving you a 36% higher chance of getting lung cancer, would you consider that clean? And so this issue prompted Yi Sui and I to do something, but it was an accidental discovery in Yi's lab. And what he found was if you take micro nanofibers, and this is a picture, an electron micrograph of this, where the scale bar over here is 30 microns. And if you expose these very fine particles, the PM2.5, to these microfibers, you find that they stick to the fibers. But if you look very closely, you see the finest fibers have the most particles, and the big fat fibers actually have the particles stuck on the edges. So when Yi was telling me about this discovery, I said, Yi, I think I know what the way this works. The particles are neutral, just like smoke particles, but these fibers are charged, and because the fibers are charged, they create strong electric fields. And just as a charged rod can pick up tiny bits of piece of paper when you hold next to pieces of paper, these dust particles, these fine PM2.5 particles, are attracted, like the piece of paper, to these fine wires, which are charged. He says, how do you know about that? And I said, you know, that seemed like a wild guess. And I said, oh, that's what I got my Nobel Prize for. We took a focused electric field, and in that focused electric field, you have the strongest electric field. And with that very strong electric field, you can hold on to atoms and biomolecules. So anyway, this material, which was originally developed for air pollution in China, for example, actually led to the founding of a company called 4C Air. And with this company, we actually have been using its people and facilities and capabilities to show how this N95 masks not only can filter out the viral particles, but how you can retreat the masks with heat, mild heat, so you can use them over 20 times. And so this again is a fabulous thing where the filtering capability of the masks is not compromised. That's very important. If you take a mask and put it in some bleach water, you'll ruin the filtering capability of the mask. And so it turns out that moist heat works. Now, I'm going to tell you about a few other applications, but I want first to remind you of arguably the greatest hockey player who ever lived. His name's Wayne Gretzky. He's not the biggest, strongest person of hockey player in a very bruising sport, but they said, what is your magic? How come you're so good? And he said, I skate to where the puck is going to be, not where it's been. And for my advice to all young researchers, think about what's going to happen in technology or maybe develop technology and look around you and where is the world going 5, 10, 20 years from today, and decide on what to work on based on that. Okay, if you look at this, this is an estimate, again, it goes to 2030 of the global supply of cobalt. And you're finding out, as EVs get better and there's a little bit of cobalt used in EVs, there's a lot of cobalt used in smartphones, but cobalt is very, very expensive. And now they're trying to get to 3%, 2%. In fact, really trying to get rid of the cobalt in EV batteries because it's simply too expensive. But it's worse than that. then nickel is also going to be too expensive. So that's point number one. Design a battery that gets rid of cobalt. Point number two, what about lithium? Are we going to run out of lithium? And the answer is no. In fact, Yi with Chung Lu, who's now an assistant professor at University of Chicago, and I and others, have developed a way of extracting lithium from seawater. Now, if you can extract economically lithium from seawater, you've just increased the available recoverable lithium by about 5,000. And because of that, this pushes running out of lithium far, far into the future, hundreds, thousands of years maybe. Now, that's not to say we still should not recycle lithium. We should, because it's yucky stuff and it's still economically better to recycle it. But the amazing thing is that if this works, we're not going to run out of lithium. Okay, so lithium is good. Cobalt and nickel are going to be bad. Now, let me tell you a little bit about something Yi did way back when. I think he reported this in 2006. He discovered you can grow little nanowires of silicon on a conducting substrate. And when lithium charges these nanowires, these things swell. In fact, they swell 2-3 times larger in volume. But because the nanowires are spaced, you can charge and recharge rapidly. And also because the lithium goes in and goes sideways. And this energy density of this anode is increased by about tenfold, the anode part. But another amazing thing is it charges very quickly. And in current prototypes already, you can get 90% of full charge in 20 minutes. But in fact, just to get to half charge, it will only take 7 minutes. And there's an expectation that we will probably get this down to 5 minutes. Now, if you have a 300-mile battery, charging 150 miles in 5 minutes means you don't need a home charging station. You don't need charging stations at work. You just go back to the service station. You run electrical lines to the service station, and as you slowly replace the gasoline pumps with charging stations, this is acceptable. And it's a fast charging battery at higher density, which will actually move the needle. This company, Amprius, this is a tool that's making these anodes. There is one thing we have to do. We've got to get the production rate up so we can actually move up to begin to supply these batteries to EVs. Okay. But like I said before, you've got carbon anodes, you've got silicon anodes. But then we said, knowing what Wayne Gretzky said, even silicon won't be good enough. And so what we really would like is an anode made of pure lithium metal. That would be the highest energy density. And oh, by the way, make the cathode something really cheap, and that's sulfur. And so we've been working on, Yi's been working on sulfur batteries for over a decade. A postdoc of mine, now co-directed by Yi and myself with some others in his group, are working very hard to make a lithium metal sulfur battery, because we've got mountains of sulfur piling up, because you've got to extract sulfur from petroleum to make low sulfur fuels. And so the oil companies don't know how to get rid of their sulfur. Now what's the problem? The problem is that in electrolytes, you have lithium bound to sulfur molecules, typically S8, 8 molecules of sulfur and 2 of lithium, or 6 molecules of sulfur and 2 of lithium. You want to keep the sulfur on the sulfur side. You don't want the sulfur to go on the metal side, because if you get the sulfur to go on the metal side, you're going to use up all the lithium. And so the challenge then becomes, can you think of a magic membrane that allows lithium to go through very quickly and keep the sulfur on the sulfur side? This is a work in progress. There's very exciting developments. We think if this works, it will be a very inexpensive separator that can be mass produced, but we'll see. Now I'm going to switch gears completely and talk about external transport in Dynein. This is a motor that transports cargoes in a neuron. So this is a cartoon of a neuron. That's the nucleus. These are the dendrites. This is a long axon. And over here are where these axon endpoints talk to other neuron dendrites. In this cartoon, inside this few micron diameter axon, there are little motors where cargo is actually used to get stuff over here in the body of the cell out to these distal axons, but also to take chemical signals from the other neurons back to this body of this cell. And the work I'm going to describe has been led by Sam Tung over the last 3 or 4 years, but there are other collaborators down below. And the motor we're going to study is called Dynein. These are cartoons of the structure of Dynein. This is one of the biggest motors, or the biggest motor known in biology. This blue thing is one single peptide chain that goes all the way to here, to here, and you see the dimensions. This is 100 angstroms. So this is a humongous motor, very complex motor. After 40, 50 years of study, the full way this motor works is still not understood. You heard a little bit about kinesin. That motor is understood. This one was not. So we decided to study this. By using some nanoparticles, we learned how to synthesize. We learned how to make very much brighter, better nanoparticles than previously, where we can dope them with any fraction of anything we want. We learned how to so-called functionalize them. That is to say, put a thin silica coating on the nanoparticles and little pieces of PEG. And these are world record thin silica. It's 1 nanometer thick silica with little pieces of PEG. And this whole nanoparticle is 12 nanometers. And so we published this work, the first version of this work in Nature Photonics. But the part I'm going to tell you about is still unpublished. And here we grow neurons over here, and they're immature neurons. So we induce the neurons to grow their axons through this little thin microchannel to over here. We sprinkle the nanoparticles over here. They go onto the surface of the axon. Nature then looks at this, and we put a proper chemical on it, a coating on it. So it says, "Ooh, yummy, I want to take this. I want to bring it into the cell. I'm going to stick motor proteins on it. I'm going to traffic it back to the cell body." So we're just riding piggyback on all the machinery of the cell. This is a top view of this cell. And this is a picture of the transport of this nanoparticle. You notice that there's no background fluorescence. It's because we use 980 nanometer light to excite the particle. And you notice this thing is not blinking on and off, and it doesn't photobleach. In fact, we can look at this particle for many hours, days, and it never photobleaches. It's completely stable. The resolution is good enough, so when we see a reverse direct, we see it actually going on another microtubule. All right. Now I'm going to switch gears a little bit and remind you about some simple physics, but it's actually not. No, it's simple physics. Okay, so consider two molecules colliding and time is going upward, and they collide and they bounce up. I take that film and I run it backwards. And so these molecules come together, they collide and they go backward. You can't tell the difference between going forward in time and going backward in time. It's because the forces are time reversal invariant. However, if you take an egg and drop it on a hard surface, you can tell which is going forward in time and which is going backward in time. And the way we explain this is because there's, the molecules in the egg look around and say, well, I want to be in the eggshell and the egg yolk, or do I want to splatter all over everywhere? Because there are many more different states that I can be in if I splatter anywhere. So we say that the disorder or entropy has increased. However, the laws of motion are still the same. So it's the laws of large numbers that make time go in a certain direction. And this was embodied in something called the Fluctuation Theorem, which was surprisingly discovered by computer simulation in 1993. and then proven in 1994. And it says the probability of running the movie forward, entropy increasing, divided by the probability of going backward in time, grows exponentially large as you go forward in time, because sigma is the entropy flow, T is the amount of time lapse, and it's normalized by Boltzmann's constant. Okay, so you can say that the arrow of time is now quantified, meaning it goes exponentially probable you're going in the right direction in time. What was amazing about this theorem is you don't have to be colliding molecules. There's a big difference between colliding molecule, pair of molecules, and an egg smashing. And so to drive this point home, a very simple experiment we did not know about when we started our work, but they dropped little fine particles in very still air, and they simply photographed the particles dropping. You know, the particles drop because they are certain, they weigh a certain amount, so we have MGH pulling it down. And by measuring how they drop, they said, wait a minute, over here it kind of dropped upward. Over here it dropped upward. Now, if you look at the slopes of these lines and say, what's the Stokes drag? Does it agree with dropping in there? And the answer is yes. So why is it dropping upward? And the reason it's dropping upward is because of statistical fluctuations, Brownian motion. And even though there are billions of molecules seen from the bottom to the top, you can actually get time reversal time to go the other way, dropping upward, even though you're averaging over many billions of molecules. So this is an example of the fluctuation theorem. For our system, same sort of deal. We have time, we have those nanoparticles you saw moving in the cell. And so in these sections where it's moving at a relatively constant rate, we're going to look at those probabilities. So if it's at time t, it's over here, time t plus tau, it's over here. We have such rich data sets because you can follow a single vesicle over nearly 1 millimeter in this cell, this axon. We say if it starts at this position over here at time t equals 0, 10 milliseconds later, oops, it can go backwards a little bit, but it mostly goes forward. By the time you're 100 milliseconds in, it's mostly going forward, but you've got a spread. The solid lines are just the fits to this statistical measured statistical distribution, and they fit a Gaussian. Well, if they fit a Gaussian, that's great. I can stick this in here. What's the reverse time? It just goes backward in time, like so. That's an algebraic thing. So I take the logarithm of this side and the logarithm of this side. The logarithm of this side is sigma t over k. The logarithm of this side turns out to be you're doing some additions and subtractions in the exponent. And what you get is a quantity phi. You take this side and it's sigma t per unit length, and you find that it's equal to twice the mean displacement distance mu divided by the dispersion, the width. And you find that this goes asymptotically to some steady state. What's remarkable about this, the very same vesicle, one vesicle going 900 microns in various segments where it goes and it stops a little bit, then goes again. Sometimes it asymptotes to levels over here, sometimes over here, sometimes over here. Gee, it looks quantized. You take 12 different vesicles, and all 12 different vesicles are quantized. In phi, well, look, what are the units of phi? This is a distance on top, this is a distance squared on the bottom, so it's 1 over distance, quantized, 0.1, 0.2, 0.3, 0.4, 0.5. What's going on? What is the meaning of the quantization? So here's the meaning. Suppose you have got a motor pulling the vesicle, but you may have other motors. So if you have one motor pulling a vesicle and it's pulling it with this Gaussian distribution, but suppose another motor gets on and starts to pull the motor with a Gaussian distribution, and the motors are acting independently, even though they're attached to the same vesicle. If one motor is allowed to move, it doesn't affect the other motor. We say that the probability of moving is the product of those two independent probabilities. If that's true, I get to multiply two Gaussians and show that I get a Gaussian, but the mean of the Gaussian is the same. But guess what? The dispersion goes down by a factor of 2. That's just algebra. In fact, if you have n motors, the dispersion goes back by a factor of n. So then you say, 0.1, 0.2, 0.3, it's getting narrower, narrower, narrower in this beautiful quantization. So we think it's 1 motors, 2 motors, 3 motors. Okay? How do you know they're independent? Well, that's actually kind of interesting. This is our cartoon of the vesicle and the motor system. I got another motor system. And if one motor pulls the vesicle forward, we need this other thing to slip. And physically, can it slip while this thing pulls forward? And the answer is yes. Because if you pull suddenly on this, if you pull suddenly on this anchor point over here, this anchor point is allowed to slip as long as this lipid fluid gets to move fast enough to fill in the space. But if you look at how fast these lipids move by measuring, let's say, the speed of sound in these lipids, you're finding they're moving in picoseconds. This thing is moving in a microsecond. So it just slips. So we know physically that these things are independent. So now we've use this to say we can actually see in real-time cells going from 1 motor to 2 motors to 3 motors. Our particles are so bright that we can get up to 5 million counts a second in a live neuron, and we don't burn up the neuron. And so in doing so, we can actually see, we can take data of the transport at the normal cell temperature, 37 degrees, or at room temperature, 22, or somewhere in between. And here you see single molecular steps in this live cell because we have these superparticles. And now if I plot the time in between steps, we cannot resolve how long it takes to make these steps. We know it's less than 50 microseconds, but we can plot the time in between the steps, and we find the so-called dwell time fits histograms that look like this at room temperature. And as you go to 37 degrees, it shrinks. You can fit this curve to 2 rate constants, K1 and K2, sequential. So it has to go through one process, and then it has to go through another process. And it turns out they're equal. This is just, again, fitting. And so we have a probability distribution. These solid lines are the probability distribution of K1 and K2. And clearly it's thermally activated. In fact, if you say that this rate is equal to E to the minus delta over KT, it fits quite nicely. So these are thermally activated rates. We've developed a very complex model for this that is actually contrary to the existing model. And it has to do with the structure of this Dynein. There are 6 so-called AAA rings, and the action is occurring on site number 1 and site number 2, where ATP lands, gets hydrolyzed. In this hydrolyzed state, this foot becomes weakly bound and gets to kind of jiggle around. It taps down, jiggle around, taps down. But then eventually when the ADP leaves the 3 site, we know from force measurements using optical tweezers, we did not do, another group did, but once this D is out here, it's deeply bound. It's a very strong force that's needed to yank this. Other people have also discovered once the phosphorus leaves ADP plus PI, you've broken ATP is 3 phosphorus, you've broken the bond, so you get a PI, and when the PI leaves, it's so-called power stroke, and it pulls forward. So, now, what are the times, those times? Remember, there are 2 rate constants, and they're equal. Well, the time it takes ATP to get on these rings is a millisecond. But remember, it's 144 milliseconds at room temperature, so that's out of the picture. It's too fast. ATP hydrolysis, also too fast. Less than a millisecond. So you're left with only 2 things. How long does it take the phosphorus to leave? And how long does it take the ADP to leave? And so, and we believe that it's the ADP leaving, and it's remarkable because let's go further. This is the step distribution that we find at room temperature at 37 degrees. This is 0 steps, and it's quantized. It goes 8 nanometers, 16 nanometers, 24 nanometers. And we just do a very simplistic fit of a Gaussian diffusion, another Gaussian diffusion, another Gaussian diffusion. Okay? This is just fitting. But what's going on here? we can say, suppose if you have an infinite amount of time at thermal equilibrium, this molecule attached to the microtubule has kind of a squishy little spring. And so and we know from equal partition theorem that KT defines the range of the spring. Okay, so we can fit that, but more importantly, we find that the data at 22, 30, and 37 degree all fit a very self-consistent model where the time for binding and unbinding, the characteristic time, is a crazily long time. It's 46 milliseconds. But, and this is just the solution to the differential equation. So this goes from when t is much smaller than gamma over kappa. Gamma is the drag, the Stokes drag, kappa is the spring constant. You get something that looks like this. And so what's going on? How could a molecule of the scale 100 nanometers, which zips around in microseconds, be diffusing in 46 milliseconds? That doesn't make any sense. Okay, it does, because we went back to the literature and said, you know, there are two binding states. When the ATPs land on this thing, it causes this stalk, this long stalk that binds to the microtubule to rotate. And there are two orientations. One in which the last alpha helix down here, which is actually binding on the microtubule, is in this state, or it's in this state. You notice how in this state it fits like a glove. So this is a weak binding state, that's a strong binding state. We went back and looked at the literature. Okay, in this weak binding state, people have actually measured how long it takes for the molecule to unbind. And it takes 29 milliseconds. So this is amazing, because then we said, because our model is saying, how do we explain 46 milliseconds? But it actually was in the literature, buried. And so the foot pops down, it's weakly bound, pops up, pops down. When it's up, it's diffusing and it pops down. Now, how long does it take to rebind? That's easy. You have a concentration, it's one molecule, but we know the volume at which it's roaming around. And from that, other kinetic measurements have measured the binding rate. So it takes about 40 microseconds to pop down, 29 milliseconds to pop up. So it goes up, jiggles around, goes down, up, jiggles around, goes down, until it decides to take a step. Okay, so that's great. So in this cartoon, let me play this again. When the ATPs are on this, it actually changes the angles. This has been measured with structural biology, EM microscopy, and we know that. We know it's diffusing around, and when it's diffusing around, it's hopping up and down, hopping up and down. But finally, it decides PI is going to leave the 3 site, it's allowed to stick tightly again, and then it does a power stroke that lunges it forward. And so that's great. But here's some more. So we continue to calculate. Okay, that diffusion constant, that model of it diffusing around, you can actually calculate the spring constant, right? You have a diffusion time, you have a spring constant, 1/2 kx squared equals 1/2 kb t. And so the spring is very floppy. But here's another dilemma, because when it's down and attached to microtubule and does a power stroke, we know that the vesicle moves in less than 50 microseconds. So if you calculate what the spring constant has to be, it has to be stiffer than this spring constant, which is orders of magnitude different than that spring constant. So how can the molecule go from floppy to stiff very quickly? First, we look at electron micrographs. This is the data. This red and blue curves are my fits to the spreading. And so we actually see this is in the floppy state, this is in the stiff state. So we have more Brownian motion in EM micrographs. So we go back in the literature and we find that in the floppy state, where this link arm is bent, you've broken an attachment. When this thing is straight, it's attached to the 1 site and it's attached to the 5 site. Here's the 1 site, here's the 5 site, and this purple thing in cartoon over here is attached to here and over here. So they're strong links, stiff. When the ATP lands on this, you break this thing, and you break this thing and you look at the structure of this guy. When this bond is broken, oops, down here, there's a deep cleft and only one alpha helix. So it becomes really floppy. So we actually have the molecular structure that says this has got to be floppy. And when it's bound in 2 points, it's stiff. Okay, there's more. And I'm going to.
So Professor Chu, we need to wrap up pretty soon, so we have lots of questions for you.
Okay. There's something else very mysterious. If you look at Q, the heat is TdS, that's thermodynamics, which is equal to the enthalpic energy in the work, but in a round trip of the enzyme, this is 0, it returns to the same state. You plug in this stuff, and you get that F over KT is equal to phi. But when you plug in the parameters for this, you find that the temperature appears to be 1500 degrees Kelvin. What the hell's going on? So we define temperature to be 1/2 mv squared equals 1/2 KT. And in trying to analyze this work, I was able to prove a very, very simple theorem, going back to the Langevin equation that says there's 2 random motions. So one is the usual random motion that gives you the KT, but if you go to the frame of reference of the motor, in the frame of the reference of the motor, you have another thing making it jiggle back and forth. So it looks like a random jiggle. You just do the exact same algebra, and you find a necessary and sufficient condition for it to be out of its thermal equilibrium. And it's easy. If the motor is really powerful, it overwhelms the statistical Brownian motion of the fluid. And that's what we see. So in very elegant mathematics, you can actually say why this thing appears to be out of thermal equilibrium as defined by 1/2 mv squared equals KT. And finally, using the same fluctuation theorem, I was able to show that there's an uncertainty relationship between the amount of heat the motor has to generate and the entropy and the orderliness. This is the dispersion in the motion divided by, normalized by the motion. In a very simple motor with just one constant, it's equal to 2 KT. 2 rate constants is equal to 4 KT, but in general, it's greater than equal to 2 KT. What does that mean? It means if you want a more orderly motor that goes like a clock, step, 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 there's no dispersion. That means epsilon goes to 0. That means the amount of energy heat has to go to infinity. You want more order, you pay for it thermodynamically. And this motor solves that. So we've been able to prove a few more theorems. Finally, I'm not going to talk about this. We can get nanoparticles into cells at will, but with that, I'll stop.
Great, thank you very much. So we have many questions. The first ones are related to the middle of your talk. Great talk, Dr. Chu. Do you think the development of lithium, sulfur, or lithium air batteries will be done soon enough to have an effect on the electric vehicle adoption projections that you showed, or is that more of a future-looking battery technology?
Well, if it's not in a car now, it's defined as the future. But we are hopeful that maybe if this works, and we'll know within a year, and by that I mean works that you can cycle 500, 1,000 times. And if that works, the things we're doing to make this work doesn't require really fancy stuff. It's actually easier to make than Eastway's lithium anodes. So if it works, it could see a very much faster commercialization.
And then can you explain more about the development of the lithium sulfur batteries and the ways that you are trying to enhance the energy density and the power of the electrode materials? Yeah,
there are 2 things. First, sulfur and lithium metal makes it higher energy density. There's no overhead. There's not even silicon. Whereas in graphite batteries, the standard battery is not. It's 1 lithium and 7 carbon atoms. With the lithium anode, it's all good to go. There's no overhead. On the sulfur side, what you have to overcome is you've got to overcome the low electrical conductivity of sulfur, and you've got to be able to have the sulfur flow freely. And the third thing is you need this magic barrier that keeps sulfur on the sulfur side. And so we think we've we have solutions to all those problems. And again, with very manufacturable materials, they're nano and micro scale materials. And so, but those actually, you know, just parsing the problem into what are the essential things that made lithium sulfur fail in the past. And once you decide, okay, these are the three things. Also on the lithium metal side, you got to prevent dendrites from forming. But the barrier, if you can barrier, can keep the sulfur on the sulfur side and be strong enough to keep dendrites from forming, you solve that problem too. So it's those combination of things that we're developing.
Great, and your lithium extraction from the sea is a great initiative. What is your take on its commercialization and cost effectiveness?
Okay, the amount of electricity is incidental because lithium sells for $10 to $20 a kilogram, and you in today's electricity prices, let's say 4 or 5 kilowatt, 4 or 5 cents per kilowatt hour, it's like 50 cents. The thing which needs to be proven is that you can cycle. It's a half a battery. So you got seawater, you take the lithium and sodium and you try to figure out a way to get lithium into this half battery, but not sodium. And in seawater, it's 20,000 times more sodium than lithium. And then you flush the seawater out, you put in fresh water, you reverse the potential, and you discharge and you drive the lithium out. And so the thing that still needs to be done is that you want to be cycling these hundreds, thousands of times. So you can get lots and lots of lithium. And there are coatings that are in the first things where you can go a dozen times or more, but you really want hundreds, maybe even thousands. But I'm Chung Lu is continuing this work, and I'm pretty confident that the nano layer coatings that protect the sodium from getting in will be found. The sodiums are bigger than the lithiums, and that's what's causing the cracking.
And then the final question before the brief panel discussion. You mentioned in your presentation that you apply upconversion nanoparticles for imaging in neuronal cells. I'm wondering if it's safe, given that upconversion nanoparticles are known to be toxic.
I'm not sure. Upconverting nanoparticles known to be toxic is, I disagree with actually. People have been injecting lots of nanoparticles, these type of nanoparticles into rats and mice. And they can be, well, huge amounts of them, and the mice aren't dying. Now the FDA is not going to approve of nanoparticles being injected into people because they won't even allow nanodiamonds to be injected into people, and that's really chemically inert. Gold particles can be toxic. And so what we found, but it remains to be seen whether FDA is going to approve, is that these nanoparticles dissolve very slowly over months. And what is left is our thin little shell. And so the materials of the nanoparticles are toxic. But if you release them very slowly and they pass through your kidneys, that's okay. I mean, we use gadolinium, for example, for MRI imaging. And there are upper limits on how much gadolinium you can actually put to use MRI imaging because you're injecting molecules with gadolinium into humans. And so, but the slow release could be acceptable. But we already know lots of nanoparticles in the mice and rats, and they seem to be, they're not keeling over. Quantum dots are toxic. But they're made of lead and cadmium and selenium and other stuff. Those are badly toxic.
Great. Thank you very much, Professor Chu, for participating in this inaugural Global Nanoscience seminar. I'm going to pass the MC over to Yi Cui.
Well, thank you, Terry and Steve, Paul, Laura, great talks. for the time consideration, maybe let me just ask one question to both of you, Steve and Laura, right here. You give great examples about what you have been working on, what you are passionate about, assuming you don't have resource limited you. What are the exciting problems you think nanoscience community should tackle? There's still thousands of people now online, right? So, and can you share with us, assuming the resource is not limiting you, what do you want to work on for the next 10, 20 years? Let's be brief. We only need brief answers, no long answer. Maybe Laura, do you want to take that first and give Steve a break? We just finished a long talking.
Yes, I feel more confident to make some comment along the line of nano-optics. So because we know that in optics, we still have the problem that conventional optical components are very bulky and they have a lot of limitations. So I have been wondering whether DNA nanotechnology especially can help nanophotonics. For instance, now in our field, there is a very exciting research direction called matter surfaces. So this topic was originally proposed and developed by Professor Capasso's group at Harvard. So the essence is that conventional optical components are kind of bulky and heavy because for natural materials, normally they have relatively low refractive indices. That means light needs to propagate substantial distances to accumulate the phase to yield a certain functionality. The now optical matter surfaces give us the opportunity to advance the field forward to really make ultra thin and flight optical components. So I have been wondering whether, especially at visible frequencies, so imagine if we have these rotary DNA-based plasmonic pixels that can be really positioned at different individual pixel positions, then by control the orientation, therefore the geometric phase of each antenna itself, then we have the opportunity to really control the phase delay at each pixel, really at the sub-wavelength level. So that is something I'm very interested in, and I don't know how long we will be able to achieve such control at visible frequencies, but I do think it is a very exciting and fun direction to play with.
Thank you, Laura. And Steve.
Well, first, I agree with Laura, but it's not only just me. Applied Materials has made that solidly in their next immediate technological thrust, not to do DNA origami, but actually to use lithographic methods. And they've shown that you could get projection of light into very flat things for total immersion. And, so instead of Google glasses where you have this stupid little camera, you can actually project into the lens itself with very, very lightweight materials. So they're marching in that direction as we speak. I think you had a glimpse of what one can do with nano designs in better batteries. I think this is a continuation of things that Yi started decades ago, but continue to make improved batteries until we do get a battery that can have 2 or 3 times higher energy density by volume and weight, so that you're not lugging around 1,200 pounds worth of battery, which is where the Tesla is today, to maybe 300 pounds, which is the equivalent weight of an internal combustion engine and transmission. The battery cost will come down by twofold, but in addition, you need this very fast charging. Again, think 150 miles in 5 minutes. You would automatically buy an electric vehicle. You don't need a subsidy after that, if their cost $25,000. And so those are big challenges. Electrochemistry is something I didn't mention. Again, where's the puck going to be 10, 20 years from today? even oil companies are saying renewable energy will be costing an average of 1.5 cents a kilowatt hour. 1.5 cents a kilowatt hour, you can say that in terms of just pure energy, and that's equivalent to how many million BTUs of natural gas in terms of energy. And it turns out that it's equivalent to $4 worth of natural gas per million BTU, which is a little bit lower, a little bit higher than the world market price. US natural gas is much cheaper because we're swimming in natural gas, but in Europe, they're paying $4, $5, $6. So that means renewable energy, electrical energy, could be cheaper than fossil energy. And so now you have to think about what can you do with electrochemistry besides making batteries? And the answer is, can you develop very inexpensive ways of making hydrogen with clean electricity? And there the problem becomes, how do you make it so you can get electrolysis very compactly, because the cost of the plant is the size of the plant. And there are many opportunities to try to improve electrolysis. Already with commercial electrolysis that we have today, it's getting pretty exciting, but there's a great opportunity to make it even better using less energy in a more compact geometry. And again, this is something that E and I have been working on for the last several years. That's again a nanotechnology. So everywhere you look, you can find places like this, but where you have to look says, where's the world going to be 20 years from today and 10 years from today? And that's where you look.
Thank you, Steve. I like your conclusion remark is to look at where in 20 years where we were going to be to find a problem to work on. So with that, let me thank all the three speakers for the wonderful talks, and also audience to stay with us asking great questions.